So first, I want to thank the organizers for the opportunity to participate in this conference and honor my friend and longtime collaborator, Richard Kirschek. We've done a lot of things together and enjoyed it all the time. So today I'm hoping to convince you that NMR spectra and molecular dynamics simulations of single-stranded short RNAs provide insight into strengths and weaknesses of force fields for predicting 3D structure. And the work has primarily been done by Scott Kennedy and Jianbo Zhao. And Scott Kennedy is next to me in case I have any trouble with my computer here. So here's an outline of what I'm going to talk about. The question is, how well do simulations with two particular force fields, they're both part of what's called the AMBER set of programs. One is called ROC RNA, that was developed here in the Dave Matthews lab, and OL3, which was developed in the Czech Republic in the Sponer lab. Sometimes it's called FF10. And so the question is, simulations without experimental data, can they agree with NMR spectra, single-stranded, in this particular case, the hexamer UCUCGU. And the whole idea is, if you know enough about the physics of RNA, then maybe you don't have to do as many experiments. So why even study single-stranded RNA? So I'm going to give you a short introduction, talk about the molecular dynamics method and results for the hexamer, and then talk a little bit about what you can easily measure it by NMR. Scott's next to me, probably doesn't agree with easily. And then comparisons between MD simulations and NMR results for the hexamer. And then I'll try to summarize. So why study single-stranded RNA? Well, in non-coding RNAs, roughly half of the nucleotides are single-stranded. And they are also potential therapeutics. Single strands are important for predicting thermodynamics of folding. So for example, if you want to predict what the free energy or the thermodynamics is going to be for predicting the duplex GC-GC, you have to be able to not only predict what the duplex is going to do, but also what the single strands are going to do. And so that's one reason why I like studying single-stranded RNA, because one of my one of the things I would be most happy about is if you could really put into a computer the sequences and come out with the thermodynamics rather than doing all the experiments, the kinds of experiments we've done over decades now. The other thing I like about single strands is there's no base-base pairing. So it focuses on stacking, on torsions, and on non-base pair hydrogen bonding. And I'll show you one particular case of that I find particularly interesting. The systems are small, and they typically change stacking in less than a microsecond. And so in the times that computers at this stage can do simulations, that allows you to see a lot of transitions. And as I referred to a little bit earlier, I think short oligonucleotides can be therapeutics in the future. So this is an example of an idea that we have, and we haven't published anything on it yet, although we are in the process of writing something up. And the idea is, can you use some kind of short oligonucleotide in order to interfere with some process, let's say for a virus? In this particular case, we've been looking at a splice site from influenza A. So there was a talk about influenza this morning, and so this kind of plays off of that. And it turns out that what our lab, particularly Walter Moss, has predicted in the past is that the splice site, which is shown here in the red triangle, where that splice site is in the secondary structure, there are two possibilities. And actually, in experiments we've done in solution, you can see that these two structures that are shown on the left part of the slide, the red triangles are where the splice site is. So the left secondary structure is a pseudoknot, and there the splice site is encased between two GC pairs. So it's not very accessible for splicing. This is a 3' splice site. The right secondary structure, S7HP, in that secondary structure, so these two are in equilibrium in solution, in that secondary structure, the splice site is in the middle of a 2x2 internal loop, so it is accessible. And so the idea is, if you could force with some small oligonucleotide the left-hand pseudoknot to be more stable so that it never can change into the right-hand secondary structure, then you'd screw up splicing. And so we made some model systems. Those are shown on the right-hand side. And I'm really going to talk about HP2, PK and HP2 HP. So what's similar between what's on the left part of the screen and the right part of the screen are the hairpin loops. So in the pseudoknot, the hairpin loop at the top of the pseudoknot is 8 nucleotides. On the right-hand side, the hairpin loop is only 6 nucleotides. So the question is, can you use a short oligonucleotide to make the longer hairpin loop more stable? So the answer is yes, you can. You can use a pentamer that was synthesized by Richard and Ella Kioshek, and you can force this equilibrium. You can force this equilibrium shown at the top of the slide to go towards the red hairpin loop. And so this just shows an NMR titration with about half millimolar of oligonucleotide at the bottom and no oligonucleotide at the top, and the RNA is present at half millimolar. And so you can get rid of the secondary structure that's shown on the right-hand side. And so the idea is, can you actually do this in a cell, such as you've heard talk about siRNA in influenza this morning? And this is just an idea at this point. This is really as far as we've gone. But I think, at least if you look at the thermodynamics, et cetera, it's possible. So this is some history of MD simulations and NMR studies being compared for short single strands. So we started with tetramers, which are shown in the first bullet point. And it was a disaster in terms of making good predictions for what they were going to do. I'll show you in the next slide, and I'll come back to this slide. So what was predicted for all of those sequences that were shown at the first bullet point, and this is just 2 of them, what was predicted is that nucleotide 1 would intercalate between nucleotide 3 and nucleotide 4. And the reason it would do that is because you could form hydrogen bonds between amino groups on a base, either C1 or on the right-hand side, A1. You could form a hydrogen bond, strong hydrogen bond, between the amino group of those bases and a non-bridging oxygen on a phosphate group. And you could form additional hydrogen bonds between terminal hydroxyl groups and non-bridging oxygens. So that's what was predicted In NMR, you did not see anything like this. So that said, okay, there's something that needs to be fixed in the force fields. So a variety of force field developers tried to fix that. So several of them shown in the fourth bullet point down here basically reduce the hydrogen bonding strength of those kinds of hydrogen bonds. One of the force fields I'll talk about today, rock RNA, did not try to change any hydrogen bonding. They just revised torsions. And they also reduced that intercalation by a lot. The other thing that reduced that intercalation, so at the first bullet point, you'll see underlined the tetramer CAAU. The force fields predicted that would be 90% intercalated in solution. It was not intercalated in solution. If you added to that tetramer a U on the 5' side and a C on the 3' side to make a hexamer, then the force fields did not predict intercalation anymore. So just by moving the terminal hydroxyl groups away from the CAAU part of the molecule, you got rid of the intercalation. So that's one piece of data that sort of tells you something is important about being able to get several hydrogen bonds close to each other. So getting them close to each other. If you put the terminal OH groups further away, then you don't predict the intercalation anymore. So now I'm going to talk about molecular dynamics for a while, and also the next hexamer that we've done. So we've only done 2 hexamers at this point. And this is not published yet, although we submitted a paper. So in molecular dynamics, what you do is you use quantum mechanics and statistical mechanics to predict what the energetics are for individual structures of a nucleotide or between nucleotides. And what's different between the 2 force fields that I'll talk about today are the torsions. So I'll show you a plot from one of the rock RNA papers. So these are all the kinds of physical things that you have to somehow mimic with an equation, such as on the right-hand side of this slide, and get an answer that's relevant to experiments. So this slide is from the paper that's cited in blue on the bottom, but it has data also from another force. That's the rock force field is in that blue citation, and the OL3 is in the green citation here. And so what this shows is what's called potential of mean force, which is essentially thermodynamic parameters like a free energy, versus angle of a torsion. And so, and this is all from quantum mechanical studies, except for what's shown in red here. So the red curves are from the database, from the PDB database. And sometimes the quantum mechanics is close to the PDB database, and sometimes it's pretty different. And in particular, it's very different if you look at the middle plot at the bottom. So that's for a torsion for guanine, monomer guanine. And in the PDB, most of the guanines are anti. In quantum mechanics, it predicts that a syn G in guanine is more favorable than an anti-G. So that's a real difference between what's seen in crystal structures and what the quantum mechanics and statistical mechanics predicts. One of the things when you're doing simulations using all these mathematical terms is getting what's called convergence. So you have to start a simulation with one structure. And what you hope is that if you started that simulation with a different structure, in the end, you would come out predicting the identical structures. That turns out to be, you really cannot prove that, which is why I put on the top of the slide, convergence is in the eye of the beholder. And this is also an advertisement for a famous sculptor in Rochester named Albert Paley. So this is one from, he has his sculptures in many museums, but one is a Memorial Art Gallery in the University of Rochester. So how do you deal with the fact that you can't really prove convergence? What you can do is you can try different starting structures. So that's what we've done, and see if you get the same result from your simulations. So the first 4 starting structures here, we've used on everything that I'll talk about today, including the tetramer. Well, yeah, everything I've talked about today. The last 2, 5 and 6, we've used in certain cases, and I'll try to remember to tell you when we've used this all 6. So this shows 4 simulations of the same hexamer, UC, and starting in 4 different starting structures. So random coil for the upper left one. The one below that, you're starting with a U6 inverted nucleotide with G5 anti. On the right-hand side, at the top, starting with A form, simple A form. And on the bottom of the right-hand side, starting with nucleotides 1, 2, and 3 stacked, and then a break, and then 5 and 6 stacked. And I'll talk a little bit about how these things match up. You don't get exactly the same answer necessarily when you start with a different structure, but you see similar confirmations all throughout no matter what you start with. You see similar confirmations produced. So we just add them all up is what we do in the end. So these simulations are from the OL3 force field. The next ones I'll show you are from the rock force field, and you'll see there's a big difference. So you see lots of variations here. So I should tell you the y-axis is root mean squared deviation versus A-form RNA. And the x-axis here is time in microseconds. And so there's a lot of variation in the time in what kind of confirmation is generated. And you'll see that's very different for for rock RNA, which is the next slide. So the previous slide, the RMSD with A-form was averaging something like 5.5 angstroms. With rock, that average is about 3.5 angstroms. So first of all, rock is always predicting something that's closer to A-form, but also many fewer different conformations. And that's illustrated on the next slide. So there's a big difference between these 2 force fields, what they are predicting. Questions are, okay, what's being done better in one force field than in the other, and sort of by playing around with the 2 force fields, can you get something that, as I'll ask later, that agrees better with NMR? So that's the question: is how well do these force fields agree with NMR experiments? So this goes back to OL3. And again, on the y-axis is the number of frames with a given RMSD, and it's the number of frames with a given mass-weighted RMSD with A-form reference. Okay, and so we've done these experiments now, both NMR and simulations, at 3 different temperatures, 2, 15, and 30 degrees centigrade. And while I have this slide up, I want to point out one thing. So the 2-degree data is solid line, and on the left-hand side of the first mountain that you see is one of the conformations that's predicted half of the time, and that actually has a C2 G5 base pair. There's no NMR data that's consistent with that at 2 degrees or at 15 degrees for that matter. So that's one thing that's clearly wrong. The second set of mountains down, that's at 15 degrees. And I'm going to try to concentrate on 15 degrees because we think that is the best thing to compare between the 2 force fields. So the next slide is going to show the results, similar results now for rock. And you'll see that the mass-weighted RMSD with A-form, first of all, it's at a smaller number, around 3.5 angstroms. And it's not, there just aren't as many structures that are generated when you use rock. So that's a very important difference between these 2 force fields. Which one is right is a separate question. And so to try to figure that out, you have to do some NMR experiments. So I'm going to say something about the kind of experiments you can do and what you can look at. So I'll talk very briefly about chemical shifts. We haven't used those for very much. I'll show you one slide. But what we've used a lot is predicted distances from those simulations I showed you compared to distances measured by NMR. So NOE, nuclear Overhauser effect distances. And also what we call absent NOEs. That is, those simulations, those molecular dynamics simulations will predict sometimes short distances between 2 protons. And if that's true, then they should generate a nuclear Overhauser effect mountain. I'll show you, a plot. And so often, not often, but sometimes we see a predicted NOE and it's not there in the spectra. And also in NMR spectra, you can get what are called scalar couplings, which are telling you what the torsions are. So that's another thing that we compare between the NMR experiments and the simulations. So just looking at chemical shifts, so this shows a plot of chemical shift versus temperature. And you don't see, what's important here is you see very little, if any, cooperativity in the temperature dependence of the chemical shift. And that's what you expect if you have a single strand rather than a duplex. So we're looking by NMR at the single strands. The simulations are single molecules, so they cannot form duplex. Unless you set up the simulation a totally different way. So when I talk about NOEs, they're from spectra like this. And so as you can see, the box in the middle, what's shown is those are really mountains. And so what you do is you measure the volume of those mountains, and that tells you from the equation that's on the right-hand side what the distance is between those particular protons. And to do that, you have to assign each of these basically mountains to particular protons. And so that's what Scott is an expert at doing. Okay, so then the question is, how do the predicted conformations compare to what you see by NMR? And one thing to be aware of in both cases is that you do not expect to have a single structure. You expect to have an ensemble of structures. So when you're doing predictions, you're generating lots of different kinds of structures. And as I showed you earlier, you get far fewer when you use a rock force field than when you're using an OL3 force field, but it's not clear which one is more correct. It's still not clear, by the way. So you have to realize that you're not looking at single structures, you're looking at ensembles when you're looking at a single strand in solution. Okay, so these are the things that I'm going to talk about. So first one I'm going to mention, which is really, it surprised us. And it's, I think, something that is very favorable for thinking about the force fields and the simulations and the molecular dynamics. And that is the G5 is predicted to often form a hydrogen bond between its amino group and a non-bridging oxygen in its own phosphate, okay, in the G5 phosphate. So, and it can only do that if the G is syn. So that's what's shown on the left-hand part of the slide. So at the top, there's a syn G, and the dotted line, and we call it a GPO hydrogen bond. That's the hydrogen bond that I was just talking about. If the G is anti, as shown in the bottom structure, then you cannot form that because the amino group is on the wrong side of the phosphate group. On the right-hand side of this slide shows again that hydrogen bond from the G5 amino group to its own phosphate, non-bridging oxygen of its phosphate. And the question is, can you see that in an NMR spectrum? And at first, we did not look for that. But then when we were looking at the simulations, we could see, if you look at the yellow dotted line, we could see that there was an NOE. So it was not something that we initially looked for or saw in the spectra. These are spectra in water. But the simulation said, well, if the simulation is right, that should give you an NOE. And it did when we went and looked more carefully at the spectra. So I think that is something that simulations can do for experimentalists. Okay. Just a minute, I have to find my pointer to change the slide. The next slide I'm going to show you is something where the simulations are qualitatively correct, but not quantitatively correct. And that's, this shows a different sequence. This is actually the sequence from the sequence that we published. But the same information and same kinds of numbers are true for the hexamer, the G hexamer that I'm talking about today. So this shows that if you have the 3' terminal nucleotide and the nucleotide before it, in A form, then between these two particular protons that are coupled by this yellow dotted line, they're like 6 angstroms away, something like that. If the terminal nucleotide is inverted, then those same 2 protons are only 2 angstroms away. What you measure in an NMR spectrum, the volume of those mountains I tried to mention to you, goes as the 6th power of the distance between the 2 protons. So if you have an inverted terminal nucleotide, you predict a huge NOE, a huge mountain in your spectrum. If it's A form, you expect to see almost nothing, or maybe even nothing, depending on your instrument. So you can tell something about how often you have an inverted nucleotide. It turns out, because of the particular NMR spectra, we can't do that very quantitatively for the G hexamer. We could do it for the previous hexamer, non-G hexamer, which is what this slide is coming from. But even with the G hexamer, if it's inverted 60% of the time, which is what the simulations predict, you should see a huge mountain. You do see a mountain, you see an NOE, but it's not huge. So the simulations in both hexamers do predict qualitatively that a fair amount of, well, the simulations predict a lot of the 3' terminal nucleotides are inverted. They are inverted, but not 60% of the time. So the previous sequence, the one that we've published, says, well, maybe from the NMR, maybe 6% of the time. So it's off by, the prediction is off by something on the order of a factor of 10. Okay, so you can predict, as I've already sort of implied, from the simulations, you can see distances between protons, and what you have to do to compare to NOEs, to NMR spectra, is to go through every frame and predict how the distance between each proton, essentially, or ones that are reasonably close, you would expect to see an NOE, how close they are, and therefore how large an NOE you expect from that. And then basically you just average that using R to the 6, okay, as shown in the top equation here. You just average that to see what you would predict for your NMR from the simulations. And absent NOE, I've already sort of mentioned to you, you predict an NOE distance. If it's short, you expect to see an NOE, and sometimes they're short, but you don't see any NOE. So that argues against your prediction, and therefore your force field. And the scalar couplings, of course, you can measure from every frame in your simulation. And you have to do that because you have to remember it's an ensemble. It's not a single structure. And then compare what you predict from the simulation to what you measure by NMR. I'm going to skip this slide. And so in RNA, typically the sugars are either C3' endo or C2' endo. In principle, all the things on the circle can be formed, but the ones that have the best energy are C3' endo and C2' endo. So that actually helps to compare the NMR to the simulations. So that sugar pucker is something that you can predict, and it's something that you can measure by NMR. And so this slide shows on the y-axis the fraction of C2' endo by either NMR or by the force fields, OL3 or rock, versus temperature. So the experiments have been done and the simulations have been done at 2 degrees, 15 degrees, and 30 degrees. And no two agree. So that was kind of a disappointment. But it's not a big disappointment for the following reason, that the difference in free energy between C2 prime endo sugar and C3 prime endo sugar is only about 1 kilocalorie per mole, which is a very small energy difference. And so I think of the sugar puckers, the ribose puckers, as kind of being like canaries in a coal mine, that they're very sensitive to what you're going to be using in your force field. So I'm going to skip this. So it turns out that, I'll go back to that slide. In the ROC force field, it likes to predict C3 prime endo, so very little C2 prime endo for most of the nucleotides, except for U6. OL3, it's more spread out. NMR, it's kind of 50-50, C2 prime endo, C3 prime endo, maybe 60-40. So that is the difference between the force fields and the NMR. For the previous hexamer that we studied, it's different. It's actually, the NMR says, oh, more C2 prime endo. Oh, I'm sorry, more C3 prime endo. NMR says more C3 prime endo than C2 prime endo. And ROC does a better prediction of the pucker in that case. So I'm basically out of time for the talk, and I want to save time for questions and for talking about Richard. So these are the kinds of things that we use to score the force field. So I'm going to go to the bottom line. Oh, what we compare is what's called sensitivity, which is in the numerator, NMR observables predicted with an error range, divided by experimentally measured NMR observables. So that's one thing that we use to compare force fields. We also use what's called positive predictive value. The numerator is the same as for sensitivity, but now the denominator is simulation predicted NMR observable. So what does the simulation predict? And in the end, we look at what's called accuracy score, which is the square root of these two, of sensitivity times positive predictive value. So what's the bottom line here? So I'll just refer you to the bottom line of this slide. And 15 degrees is the best comparison. And that's because OL3 at 2 degrees has a GC pair, which we know does not exist in an NMR tube. So for accuracy, even though it has some GC pairs, even at 15 degrees, about 15% GC pairs in the simulations, the accuracy turns out to be 86% basically, using all the NMR characteristics I talked about a little while ago, a couple of slides ago. And ROC, it's about 67%. So you say, okay, in that case, then it looks like OL3 does better, even though one of the things it predicts is just not true. For the other hexamer that we've done and now published, it's cited at the publication is cited at the bottom, it kind of comes out the opposite way. So if you just look at the left two columns, the OL3 predicted the NMR spectra roughly 67%, and the ROC predicted the NMR spectra roughly 75%. So each force field has things that are right and things that are wrong. And the question is, how can those force fields be improved? And so one of the things that we like to say is that the experiments on single-stranded RNAs are one case that's good to compare your force fields against. So force fields typically are compared against very structured RNAs. But I think that you want to compare predictions against as many experimental results as you can. And so we think that comparing against single-stranded RNAs is one of the criteria that should be used in thinking about force fields. And also they can tell you something about, okay, this particular part of your force field, let's say, your sugar pucker is a problem. So these are the people who helped us on the left. And on the right, and Kyle Berger helped us with the initial hexamer that we studied. And Jianbo Zhao and Scott Kennedy are the people who did the real work on the G hexamer that I talked about. So I'm going to think that is all I'm going to say, because that's all the time I have. And I'll take questions, but then I'm going to tell you about some of my experiences with Richard.
Professor Turner, thank you very much for your interesting lecture. And now it's time to questions. Any questions, Professor Ryszard Kierzyk?
Hi, Doug, it's me.
Okay.
In the past, when Dave was running these tetramers, yeah, he
Yes.
was often like tetramers with RNA inside, or with some, for example, a bromo, where in that case you have somehow some nucleotides where they structurally fixed. So is it possible?
Yeah, wait, Richard. I think you're confusing our tetramers, which you did help us with, but we never tried any bromo. I mean, I think that's a good experiment. We just never did it.
Just RNA was that case.
Just straight RNA. But you're giving me a chance to say one thing, which I think I said earlier, but I want to stress, is that the real long-term goal in this is to get from quantum mechanics and statistical mechanics, which can be done on computer, get predictions that are good enough so you don't have to do experiments anymore, or at least not as many experiments. And so it's not close to that yet, is the bottom line here. But it is a realistic goal, I think. And as computers get faster and faster, you can allow these simulations to go for longer and longer. And so that's important in getting convergence, for example. So there's a lot left to be done, I think, is maybe the bottom line of this talk.
So I remember when we talked many times before, you always said, when I'll be retired, I will be honorable postdoc, and Dave met you and work on this huge calculation machine to predict structure. So it looks like his dreams have come true already.
Yes. So, in fact, just ordered, I hope, this last week, because I didn't do the ordering, a staff member did the ordering. I ordered a $7,500 computer, which I'm going to do simulations on. Okay, so I'm hoping to learn from Dave, and probably mostly from his students. I'll have a desk in an office with 3 of his students. I'm hoping to learn how to do simulations myself.
Okay. So my last question is, when you plan to visit Poznań?
When I, as soon as I feel safe going to Europe and going through airports, and I'm looking forward to that. And so is Joanna.
Always.
Joanna will come with me. Joanna knows more about Poznan than I do, and I think maybe even you do. I think she's visited one of the museums that you've never visited.
Yeah.
The last time that we came together.
Okay. So, thank you.
We have one question from online audience. So if Professor Adamiak want to ask what cations and anions were used in water simulation box.
Yeah, that's a good question. So we use sodium chloride. And actually we really, yeah, so we don't use potassium, and at some point probably we should. But that's a good point.
Is other question? Paweł Zmora.
Hello, Professor Turner and Doug. It's nice to see you.
Nice
I
to
have
see
several
you.
questions.
You're very small.
I'm wondering how your predictions are relevant for our biological models, because if I'm correct, you checked everything in some chemical solution. Can you include in your mathematical modeling some factors from the biological cells to be more relevant for biologists?
So I think that that they are relevant, and one reason is because so the one thing that is far in the future probably. Well, some people are trying to do it. Is okay. Can you sort of worry about what proteins are doing, right? So protein force fields are actually very good already. It's the RNA force fields that need to be improved, in my opinion. Another thing that people worry about is, we use sodium. Typically, we don't use magnesium in our experiments. And in our NMR, we've never used. Well, I guess we've never used magnesium in our NMR. But I think that what people don't realize is that the interactions in RNA are very local and very strong. The hydrogen bonding, like a GC pair, you have 3 hydrogen bonds. A single GC pair can increase your stability by your equilibrium constant for forming a duplex. Let's say by single GC pair, you can get a factor of 100 in an equilibrium constant, for example. So the local interactions are very strong. The other thing that people often ask me is, so our thermodynamic experiments that we've done, and we're trying to model now with the simulations, the thermodynamic experiments, we've always done at 1 molar sodium chloride. And people say, well, that's not what's in the cell. In the cell, it's a lot of potassium, a lot of magnesium, a lot of everything, right? But what people don't sort of realize, I think, is that when you have, in particular, a large RNA, you have what's called counter-ion condensation. And what that does is no matter what your bulk concentration of sodium or potassium or whatever, no matter what your bulk concentration is, the local concentration around your double helix is higher. It's on the order of, have to think about this a second. For RNA, where the negative charges are pretty close to each other, closer than in DNA, it's something like, it's close to 2 millimolar. 2 molar, I'm sorry, 2 molar. It's close to 2 molar right around the RNA. So and everything, the sort of local environment is really what counts in terms of what structure is going to be formed by your RNA. So I think the experiments are actually more relevant than most people think to what's in the cell. Can I talk about Richard now?
Yeah, of course.
Okay. So I have to find, I have to be able to change my slide. So does that mean, can something be done so I can change my slide now? So change my slides.
Can we ask the technician to do the presentation?
So should I open the
Okay.
presentation?
You should be able to change it.
Yep. Yeah, there we are. Okay.
Okay.
Thanks. So, yeah, I wanted to have enough time to talk about Richard. So we met in, and correct me if I say anything wrong, Richard. So we met in 1985 when we were both on sabbatical in the chemistry department at the University of Colorado in Boulder. And we were just lucky, A, to be there at the same time, and B, to be in labs that were opposite each other across a hallway. So I was in Tom Check's lab, and Richard was in Marv Carruthers' lab. And we just happened to meet in the hallway. And we started talking. And, you know, we started talking about RNA. You know, we didn't even realize that each of us worked on our RNA, really. And so my lab at that time had been synthesizing RNA using biochemical methods, in particular, you know, one developed by Aki Uhlenbeck. We were making 1 hexamer in a month. Richard told me he was making 4 hexamers a day. I immediately saw this as transformational for thermodynamic experiments on RNA. So I got to know Richard really well. And the next slide shows, so this is Richard telling people who had come from my lab and were in my lab and sort of came back to celebrate my living to be 60 years old in 2006. And he's showing them how RNA synthesis was done by him at that time in 1985 and then for many years since. So the next slide then oops, now it works. Okay, so now the next slide shows me with Richard in Marv's lab. And you can see his flasks are set up and he's synthesizing 4 RNAs a day. And so we then published, so this was 1985. We then collaborated for a year or so, just for 1 year, and published 6 papers in 1986, one of which has been cited over 1,000 times. So Richard's chemistry was really transformational. And I know there are other people in Poznan who also contributed to that chemistry. So I think Poznan has done great things for the RNA world. So then my next slide, let's see. Yeah. Shows us accepting an award from AAAS. So AAAS, the American Academy for the Advancement of Science, has an award for Poland-U.S. science collaborations, and so we won that award, and this is we're accepting it here in Warsaw. And what you can see, one of the reasons why I'm showing this slide is that Richard has hardly changed in the 35 or 36 so years that we've known each other. And I've changed really a lot. So my next slide shows Wooch. So this is a picture of Wooch. And so there are a couple of times which I'm going to mention. One is illustrated by this slide, where this collaboration almost died. And the first time was the first time that I visited Poznan, which was in November of 1985. Richard kindly set me up to also visit WUCH, the chemistry department at WUCH. And he put me on the train and had arranged for one of the professors in the chemistry department at WUCH to pick me up on the platform when I got to WUCH. And I had both feet on the train, and I looked down at Richard, and I don't know why I asked this. I just said, "Oh, how do you spell WUCH?" And he looks at me, he says, "Oh, it's L-O-D-Z." I'm like, in English, you would pronounce that Luds. I would never get off at Luds if I was going to WUCH. And so if I had not asked that question, I have no idea where I would have wound up. Someplace in the south of Poland. I don't speak any Polish, even now. It was behind the Iron Curtain. Probably would have been arrested as a spy or something. I have no idea what would have happened to me. So that's one case where this collaboration could have evaporated very quickly. The next slide shows another example where it could have ended very quickly, but much later, fortunately. Yes, much later. And I just picked a slide with a gel on it. And those of you, because this is about running gels, and this is mostly for the students in the audience. So Richard, if you've ever watched Richard work in lab, he's got incredible energy, he works very fast, he's often doing two things at the same time, and he's getting an incredible amount of data any day, any one day, or make an incredible amount of RNA if he's synthesizing something. So one day when he was on sabbatical in my lab, he was running a gel of oligomers, and he likes to get as much data out of a gel as possible. So he runs two sets of samples on one gel. And so this one day, he loads a set of samples, lets the dye run down about halfway down or something like that. And then he goes to load a second set of samples. But he did not turn off the high voltage. So the high voltage went into his hand and fortunately came out at his wrist. It could have, kept going and maybe the collaboration would have been over. That would have been a real disaster. So I just mentioned this to students that, yes, you should, work as hard and as fast as Richard does. There's a lot to be learned from the way that Richard works. But sometimes you can try to be too fast. Okay, and then my last slide. Shows my son, Ricky. And the reason I show this slide is because when we were about to have a child, my wife and I, we didn't know if it was going to be a boy or a girl. So we chose names for a boy, we chose a name for a boy, first name for a boy, first name for a girl. And we wound up having a boy, as you can see. And the name that I first said to Joanna, my wife, was, let's name him Richard after Richard. So that's the English form of Richard is Richard. And she thought about it for a few seconds. And she said, yeah, that's a good idea. And she also came up with other Richards that had been important to us. So actually, Ricky, the name Richard was inspired by Richard, but then it was supported by two other Richards. One is Richard LaDuena, who's one of my college roommates, And he was the person who, when he learned that I was going to University of Rochester, he suggested I look up Joanna Olmsted because she was also coming to Rochester the same month that I was coming to Rochester. And he knew her from meetings because they both worked on microtubules. And so he was the person who, said I should look up Joanna, who is now my wife, after 44 years or whatever it is, a long time. And then the other Richard was Richard McIntosh, but he's called Dick McIntosh. So actually, my son is named after 4 Richards. That's what's on their birth certificates. But they're called by 4 different names: Richard, Richard, Dick McIntosh, and Ricky. So I just find that sort of amusing, I guess. So that's all I have to say today. I think I kept you from dinner by 4 extra minutes.
Professor Turner, we thank you very much again.
I thank you. And have excellent dinner.
When I was in Rochester, I was working on RNA synthesis on polymer support. And next door, it was a long hallway. On right side was Caruso's lab, on left was Tom said. That office lab was across. He was working with Elisa, I don't remember Elizabeth, I don't remember her name, and Brenda Bass, I believe. Very often you can hear very noise loud, because Doug is laughing very loudly. So even he was two floors below, you know that he is in lab, yeah. and really, that really, when we met first time, it was that situation that I was working, because that time Tech worked on IVS splicing, yeah? And he was interested in synthesis of this kind of L-19, something like that, was 15-mer RNA. It was very long RNA. And I made that, they protected, and give to Art Zauk. Art Zauk was the best technician in the world, yeah? And Art, I was at the protected, I ran this kind of, I labeled ATP, so you run gel, and Doug was sitting in his office. It was kind of big room. There was many desks around, and I was talking with Art and showing him how it looks like this gel, and Doug was sitting, and so he realized that we're talking about RNA, and he asked, "What is that? What is that?" Yeah? I said, "That is RNA. How you get this RNA? How you get this RNA?" And he told him that he is from Rochester and he's on sabbatical. At that time, I didn't know what means sabbatical. This word means. What is sabbatical? Yeah, and so after a day or two, he gave me a lecture about thermodynamics. I was near neighbors. So my first, because in near neighbors of RNA, you have 10 parameters, but you should have theoretically 16, yeah? At first time, I didn't know why is 10 instead 16. That when my first success was when I myself explained why it's only 10, not 16, yeah? That was mine. And then a few days later, he wrote such a long list, or looking at what he need. I said, my boss is Mart Carrozes. Go to him. And he went to Carrozes, and give his list, talking blah blah, talking together, yeah. And after a week or two, no response from carousers. So we decided go ahead and do this e-commerce, and that's it. Because when Doug was in Rochester, actually in Boulder, his lab in Rochester was run by Susan Freyer. She was postdoc at that time, yeah? And Doug told me that her husband asked her, do you know when this Polish guy will go to home? So you can be at home at regular time? So because I synthesized these oligomers, yeah? And Doug sent these oligomers to Rochester, where Susan and few others students or graduate student postdocs did these experiments, so they stay a long hours at the lab and instead be home. So Susan husband asked, when he be back home, yeah? I was in Rochester in Boulder 6 months, yes. Well, it was like November up to May, so that is what I always remember. You know, I remember also when Doug said one day, could you teach me how to make synthesis? I said, oh, go ahead, yeah. He said sheet on the hood was like for funnel, it was really funnels, yeah. For funnels, because on each funnel you can make one oligomers, yeah. And he was, using these funnels, the syringe, and going slowly. And as usual, it took me like 45 minutes, one step, yeah? So in his case, it took around 2 hours, yeah? and after, he was like half of synthesis, he said, you go better, you do finish synthesis, otherwise you'll be stay here overnight. And so that was our collaboration. Yeah. So, together we published around 60 papers together, yeah? Starting from this paper in 85 or 86, something like that. So for me, it is around 1/3. My publication was that, yeah. For him, for example, much less, but it's still a lot.
So what Richard said is completely accurate. So I was going to actually say some of the same things, only I ran out of time. So Richard, I'm glad you said it where I ran out of time.
Yeah, so it's like so it was good time, very nice time.
Absolutely.
Because for me, I was at that time. I was 6 months in Rochester, and I published around 10 papers, including two or three papers as first authors, yes. So, but we stay at that time at the lab like between 8 and 10 p.m. It was, Tang. It was also some guy from China, Tang, yeah. He was, so we stay whole day, very long hours in the lab. So we leave this Boulder the same day, but he went to Shanghai, me to Poznan. But so it's how it started. Yeah. Now we are a little bit older, but still young.
We hope you will visit Poznań again on Łódź.
See, that was presumably in Poznań like I believe 5 times, 6 times, something like that. I was in Rochester. I spent in Rochester around 7 years. The longer piece, it was 3 years, then was 1.5 years, and then was like many pieces, like 3-4 months. Yeah? It's this. So I know mostly everything in Rochester, every single corridor and whatever. Many people. I feel almost like home in Rochester. Yeah. In Pittsford, because Doug is living in Pittsford. Yeah, he's not living in Rochester, he's Pittsford, Stone Road.
So, okay. Professor Taranev, thank you very much again.
So we're waiting you See you.
Yeah, of course.
Presumably in 1 year or half year will be Poland will be the entire world will be a safe place to be. So we are always very welcome here.
Well, both Joanna and I have gotten the messenger RNA vaccines twice. So two shots.
So most people on the ODS is also vaccinated at least mostly twice. So everything looks okay and safe. But people just most scared to travel.
So I don't know if you're aware, but at least the Moderna vaccine, they use the thermodynamics, some of the thermodynamics that we published together. They use that to predict what will be the most thermodynamically stable sequence, and that's the sequence that they use apparently for the vaccine. So it's a little bit more complicated than that, but you may remember John Santalucia in the lab, Richard.
Excuse me?
Richard?
Yeah. I was talking a second. I was listening to her. She said because people are waiting for dinner. So.
Okay. Well, let me just mention one thing very quickly. So they use our thermodynamics of these predictions, and Dave Matthews has helped them to decide how to do it, and John Santalucia, I think you overlapped with him at some point in the lab. He has measured thermodynamics for, I think, the 2-methyl pseudo-U. So John started a company called DNA Software. John started a company called DNA Software.
Yeah.
And if you look in one of the PNAS papers from Moderna, that company is acknowledged for doing the thermodynamic experiments.
Yeah. Because we know that in vaccines they use 1-methyl pseudouridine for Pfizer and Moderna. So you presumably we can, because we know. You can talk with Dave Matthew because we will make this pseudouridine. We will get this.
Oh yeah.
We get this.
Yep.
So thanks a lot, and it was
I wish I was there.
Yeah.
But I'll be there sometime in the future.
Okay, so you are always here, very welcome.